So I want to thank everybody for joining me today on this webinar about XGal-3, Eliaz Therapeutics, and the role of galectin-3 in critical care. The webinar today is a little bit different as it will tell a lot of my personal story over the 10, 12 years, a little bit about my discovery, and the potential for XGal-3 to really transform the critical care, the intensive care unit, and really finally offer a treatment for sepsis, the #1 killer in the world. Today's webinar is on XGal-3, a life-saving solution for sepsis and for acute kidney injury. And this is just a disclaimer because we are discussing investment. We always need to have a disclaimer. So I want to start by talking about sepsis. It's surprising how many people are not aware of how devastating is sepsis. Even doctors sometimes are not aware of what a problem it is. So in the USA alone, 1,000 people a day die in the ICU from sepsis alone. More than this die from sepsis and acute kidney injury. This is only in the USA. But what's stunning is the level of the unmet medical need. There is no FDA-approved treatment for sepsis. All the treatment for sepsis are supportive treatment, antibiotics, fluids, trying to keep the blood pressure and trying to hope that the patient recovers before the septic process damages the organs, causes multiple organ dysfunction that leads to devastating morbidity and mortality. So really in the last 25 years, some will say in the last 30, 35 years, there are no real new effective therapies introduced for sepsis. We are talking about 49 million cases a year globally, 11 million deaths, 20% of the global mortality. More people die from sepsis than from all cancer put together. That's how dramatic it is. Sepsis and acute kidney injury puts a great burden on the medical system, a great financial burden. It costs over $62 billion a year in the USA alone to treat sepsis. It costs additional $24 billion to treat acute kidney injury, and both are the leading cause of readmission into the hospital and 40% of ICU costs. Now, these costs do not include the long-term consequences of sepsis. Sepsis is a devastating condition with mortalities between 20 and 40%, but in severe sepsis, we're looking at 60% mortality in the ICU, 60, which means out of 10 patients that come into the intensive care unit with severe sepsis, and remember there are a lot of them, 6 out of 10 are not going to get out of the ICU. And the ones who get out, many of them, their life will never be the same. It is a long-term sepsis, post-sepsis syndrome. They will have multiple organ damage. Many of them will end up having dialysis. Their kidney function will never recover. About 30% of them will be readmitted in the first months, but 20 to 40% of people who survive sepsis will die in the first year after the sepsis incidence. So this is to get you a sense of how big is the problem with 2 million sepsis cases a year, 4 million acute kidney injury. Sepsis and kidney injury extend intensive care unit stay by 50% from a cost point of view and from a health damage point of view of every extra day that you stay in the ICU. So worldwide, it's a huge, huge financial burden, a $2 trillion market. The serviceable, addressable market is about $200 billion a year, and the one that is really reachable, even if it's 5%, is a $10 billion market. So this is how big of a problem it is. Now, what is Eliaz Therapeutics offering and why I'm so excited about it? I'll talk later on about the role of galectin-3 in acute illnesses. I'll share with you some of my research over the 10, 12 years that I didn't share until now. So really recognizing the importance of galectin-3, we really developed a selective filter called XGal-3, that works together with common apheresis system. So it selectively removes, it has a resin and then proprietary antibody that we developed, and it selectively removes galectin-3 from the plasma. In our studies ex vivo, we show that when we run plasma with galectin-3 through the filter, when it comes out, there is undetectable level of galectin-3 through very large volumes of plasma. So it selectively removes galectin-3, so we can rapidly reduce the levels of galectin-3. It is easily adopted into common standard apheresis machines. It's a proprietary process that we have 60 patents in 34 countries all over the world. And it's really a platform indication. Why is it a platform indication? Because as many of you know who heard me before, galectin-3 drives many diseases. So we are starting pinpointed focus on sepsis and acute kidney injury that is caused by sepsis, sepsis-associated AKI. But then we're going to move to chronic kidney disease, about 15% of the population, enhancing cancer immunotherapy, and liver and lung fibrosis, as well as other indications. So basically, just to explain how it works, you pull the blood. The platform, we use a Terumo Optia. We have a collaboration and commercialization agreement with Terumo for many years, which allows us in the future to be present in different countries. We separate the cells from the plasma. The plasma gets filtered. Only galectin-3 is pulled out. So it's a very elegant treatment. It comes back, gets reunited, and comes back into the person. So the treatment can really be applied at any stages in sepsis. So really what we are doing, we are combining a well-known procedure, therapeutic apheresis, which is now more acceptable after COVID, combining with our unique anti-galectin-3 antibody, and this is Eliaz Therapeutics XGal-3 selective removal. It's a well-known technology to separate compound from the plasma. We use proprietary binding antibody. It fits widely available standard apheresis, machines widely used in ICUs, unit and in hospitals, and suitable for different indications. So let's now dive a little into galectin-3 and see why and how I came with this idea. So some of you, hopefully many of you, are familiar with the survivor paradox. And when I talk about the survivor paradox, what protects us is what injures us and gets us sick and shortens our life. We talk about the immediate response being the sympathetic system, really automatically immediate. But then very quickly after, the biochemical response by having alarming biochemical compound. And galectin-3 is a leading one. It really starts this cascade of acute response, and then there is a downstream effect. So if we look at this diagram, we can see galectin-3 in the middle. And then when galectin-3 gets stimulated by stress, by injury, through fibroblast, through macrophage, or directly, it activates macrophage to be inflammatory, and then we get the downstream of inflammatory compounds and cytokines. This is the famous cytokine storm. Or we go through the fibrotic pathway, transgrowth factor beta 1, fibroblast collagen. The end result is similar. Tissue fibrosis, organ dysfunction, death, and chronic illnesses. However, so what happens, how does it happen? Galectin-3 is a monomer. When it's triggered, it binds to different carbohydrates. It turns into a pentamer. It can drive the different ligands to the target tissue, and it creates a coating, a lattice formation. It creates a microenvironment, changes the receptors on the cell, and changes the cellular metabolism. So with this in mind, everybody initially was focused on cancer with galectin-3. Then I discovered that blocking galectin-3 attenuates inflammation and fibrosis. I got patents for it many years ago. And then the world a few years later discovered it, and research moved into inflammation and fibrosis. I also discovered that galectin-3, when you block it, you can attenuate immune dysfunction. This is already in 2005. And then when it came to me, I realized I need something dramatic for this deadly conditions that killed my father from immunotherapy in the 80s in half an hour in Cornell University Hospital, and caused immunotherapy to stop actually for many years. It was a bigger success, but he also died within half an hour from this inflammatory reaction. So my whole concept was, when you remove galectin-3, you will shut down the process. And this was also the title of the topic of my first NIH grant, where I proved in an animal sepsis model, galectin-3 comes before interleukin-6. When I block galectin-3, interleukin-6 does not go up, kidneys do not get damaged, and the animals don't die. So here you can see the very prolific role of galectin-3 in so many diseases. Our focus is on sepsis and acute kidney injury. Really, the biggest killers that so many people are not aware of, but so many people know someone in their family who went through this. So how does XGal-3 works? It disrupts the damage of galectin-3. Galectin-3 gets released into the bloodstream during sepsis and AKI very early on. It drives inflammation, fibrosis, immune dysregulation, causes organ damage, and death. It's also a very good prognostic indicator to what is going to happen. I will show you stunning data about the very large numbers, very good scientific papers, about the importance of galectin-3 levels at the day of admission to the intensive care unit in determining who is going to die and who is not going to die. So what we are doing, we are disrupting this process. And because galectin-3 is an upstream molecule, in severe sepsis, it doesn't go up 100 times, like C-reactive protein will go up from under 1 to 100. Interleukin-6 will go from undetectable to 10,000. These are downstream molecules. Trying to treat them, it's like stopping a waterfall with a bucket at the bottom. We are stopping the waterfall. So galectin-3 will only go up by 50%, will double, and yet it will have a devastating effect. And this is really the basis, the logical basis of this promising treatment. And then, of course, we have proven it until now already, and that's why I'm going to share this with you today. So once we get established in sepsis and acute kidney injury, we're going to move forward to chronic kidney disease. 37 million Americans affected. Many people are not aware of it. Almost $100 billion annual expense. And then as an adjuvant to immunotherapy, I'm back home, reducing the inflammatory side effects and enhancing the results and other fibrotic conditions. So this is our roadmap. So why does this treatment have a competitive advantage? Why, after I invested so much time and money and effort, I'm now sharing it with others, asking for the help to join me in this? Because it has a quick effect, literally within minutes. It is economic. It's cheaper than very expensive biologics, and it can be repeated as much as needed. It is well patent protected, cannot be copied. It has a long-lasting effect because you can always do another treatment. It is safe because the treatment is outside the body. There are no byproducts, there are no metabolites. You do the treatment, you remove the galectin-3, and you stop the treatment. The body still produces galectin-3 where it's needed. That's also in sepsis why you need to repeat the treatment the next day, because the process keeps on producing, and we have shown very interesting data about it. And it's profitable and easy to get to market because it's not a drug. The probability of a medical device to be successful at the stage we are now is dramatically higher than the probability of a drug, where a fraction of the drugs get approved. So what did we achieve until now? We have a very important partnership with Terum Obesity, a renowned global leader in apheresis, which is present in close to 200 countries. We have completed preclinical trial and clinical observational trials and diagnostic trials. We are preparing for a GLP study, and that will allow us to submit for clinical trials. We are backed by 6 compelling scientific publications that we have done, including a very important one that we are just about to submit. We have 60 global patents across 4 key families including groundbreaking patents in primary international markets. And we are about to file a new breakthrough designation application with the FDA. We're not guaranteed to get approved, but we have very high probability. And we're really very close. We're actually filling right now the final prototype for our animal studies, and we'll be ready to use this prototype in clinical trials. So let's step back now and see. I explained to you the logic of galectin-3 in critical care. Now I want to show you how powerful is a substantiation of this. So this is a landmark study in critical care, one of the best peer-reviewed journals in critical care medicine from 2022. The FROG study, 2,076 patients coming into the ICU. Not all of them sepsis, about 30–40%, other very extreme conditions. And we are checking the level of galectin-3 at the day of admission, okay? And we want to see what is the outcome. Which of these patients will develop MAKE, which is major adverse kidney events? Which of these patients will develop AKI, acute kidney injury? And which of these patients are going to survive and which are going to die? So you look at the convincing differences. It's a very, very high significance for people who know statistics. This is under 0.001, okay? Between the level of galectin-3 of those who didn't have MEC, average 16.9 compared to 30.2. And you can see that the overlap between the group is barely existent between the 25 and 75. This means we have a much easier cutting point in the intensive care unit, especially with an automated galectin-3 testing available through very common platforms in hospitals. And same thing with acute kidney injury. And also same thing with mortality. Look, the people who survived, the average galectin-3 a day of admission was 18.8. The people who did not survive, 29.3. You see, we don't need to remove a lot to make a huge difference. That's the promise. And the numbers, of course, are supported. About 40% of the patient developed MEC, and the 30-day mortality of the whole group was 22%. So let's now dive a little bit deeper into the science. When the investigator divided the patient into 3 groups of galectin-3 between 5.2 and 15, 15 and 27, 27 and 114, there was a striking difference in mortality. 10.4%, about 20%, about 37%. Right? All of this when galectin-3 basically is a little bit more than doubled. Look at the difference, almost fourfold. But now they took patients who had no acute kidney injury during the hospitalization and had no, galectin-3 was low, less than 10% mortality. They took patients who had elevated galectin-3, but acute kidney injury was not present, almost 32% mortality. When they took patients who had acute kidney injury, a major cause for morbidity and mortality in the ICU, but galectin-3 was low, their mortality was lower than patients who had only elevated galectin-3. And when you have both of them together, you're reaching a little bit over 33%. And very striking data on the level of galectin-3 and the severity of AKI. No AKI, and then KDIGO 1, 2, 3 are the 3 categories of acute kidney injury. When you get to KDIGO 2 and especially 3, mortality skyrockets, and morbidity, long-term morbidity, the need for dialysis, long-term goes up. And you can see how galectin-3 is involved with all of this. So you can see the basis for this intervention. So we ourselves did 2 studies. Before I'll talk to you about our apheresis studies. One was with patients coming into the ICU after cardiac surgery. So they had a heart-lung machine. They come to the ICU, and during the process of heart-lung machine, the circulation gets shut for a short time, and you cause something called perfusion injury, reperfusion. No blood comes to the tissue, and then the blood comes again. And you produce about 1/3 of the patient gets AKI. You can see how strikingly the level of galectin-3 at the time that they were admitted from surgery to the ICU. They don't have AKI yet. They just finished surgery. You can see how the levels were so different between the people who developed AKI and did not develop AKI. It was really an early indicator who needs to be treated. And when we looked at sepsis patients, same. Almost half of them had AKI, and you can see the striking difference between the group. And what I'm not showing you, that also the people who died from sepsis also had much higher levels of galectin-3. So with this in mind, I want to take you through our journey of proving that this treatment works. So we started with the Porcine study proof of concept skin inflammation model with Dr. David Sachs in Mass General Hospital, Harvard. David Sachs is really one of the fathers of immunology. He discovered the XGal-3, the self-recognition system. His life work is on xenograft implants. And so we used his model. Then we produced human data, some which I showed you, and some new one that I'll show you in a moment. And then we did 2 proof of concept specifically in sepsis. One in septic model in rats, and one the landmark study, the large animal LPS-induced sepsis in pigs. So what we did in Harvard is we prepared the animals for the study. After 3 days, we took labs, and then they got this injection to the skin of a substance called complete Freud adjuvant. It's an attenuated bacillus bacteria that can cause TB, and it causes inflammation. And the way it was injected actually causes ulcers. And then the animals either got hemapheresis or got the galectin-3 antibody removing the galectin-3. And we showed a clear difference in the level of galectin-3 during these 28 days. Actually, we went all the way to 33. But what we saw that was not part of the plan is that when the animal had these injections, their blood got very coagulated. And when we did the galectin-3 apheresis, the blood was thinning out as the treatment was going on within hours. And that's when I had my first click. Oh my gosh, this can help sepsis. So I want to show you the profound difference on day 10 between the control animal and the lymph congestion in the tissue, and between no ulcers being developed. This is just for the biopsy, and the tissue being pretty much normal. So then, on the diagnostic level, this we just presented in early 24, we looked and we showed that in normal population, galectin-3 in control is much lower than the level of galectin-3 at the admission for sepsis to the intensive care unit. What we also showed that I'm not showing today is the dynamic changes, that if galectin-3 goes up after 3 days, the patient have a greater chance of dying. If galectin-3 comes down, it actually, the outcome is better. And we are now doing more studies on the dynamic treatments of galectin-3. So these are our 2 important proof of concept. First, we had to develop a special antibody just to pull out galectin-3 from rats. It was a whole process. We developed our own antibody to be able to do the study. We used a process called single ligation and puncture. It's the most acceptable abdominal sepsis procedure. And we tested on 19 animals. 9 got hemapheresis without the column was empty, and 10 got the real anti-XGal-3 antibody. Only one out of 9 animals survived in the hemapheresis, 9 out of 10, so 90% survival compared to 11% survival. So now we come to the main study. Now, I apologize, I can't show you the really dramatic details because we are submitting to a very high impact journal. We're not allowed, but I'll show you the principles and the summary. So this is septic shock induced by intravenous infusion of LPS, lipopolysaccharide, the molecules, the chemical structures that causes sepsis symptoms, and maybe track back. People know sepsis as infection in the blood, but that's really not the new definition of sepsis. The new definition of sepsis is the body's response to the infection, the immune dysregulation, the inflammatory dysregulation. This is what sepsis is, and that's why there's no treatment for sepsis. So the animals, after preparing and putting the catheters for the treatment, they are dealt with just like their intensive care unit. 5 intensive care doctors do this procedure. They're intubated, they get fluids, they're all ready, to be resuscitated. You put LPS 2 microgram kilogram hour for 10 minutes, for 20 minutes, patient blood pressure drops, you stabilize them. So they survive the basic procedure. And then you accelerate them. Within 45 minutes, they're in full-blown sepsis. It's an aggressive model, 70 to 80% mortality in 24 hours. We start apheresis at the accelerated phase when they're already equivalent to already crashing in the ICU, 35 minutes before stopping it, and for another 3 hours. And in addition to monitoring the animal and blood pressure, they actually put a special swan gauze to check extra lung water index, pressures in the lungs, level of lactate, animals amount of vasopressors to keep the blood pressure, IV fluids. The whole thing is even there in the ICU. So I want to just show you the summary of the results. So out of 31 pigs, in the 15 control, only 4 survived, less than 30%. In the active group, almost 70% survived. And what we found, that in those who didn't survive, it was because we didn't have our final column yet. It stopped working after 2-3 hours. When we put 2 columns in a row, the effect came back. And now our new column that we are testing is about 10 to 12 times the capacity of the columns that we tested here. You always learn from where you're not as successful. So basically, the sham group exhibited a rapid decline, but most important is that we saw already within 35 to 45 minutes, an hour, less than 1 plasma was filtered, and less than 40% with about 40-50% efficiency. Now it's much higher. We saw that the septic process, the effect on the lung, the acute respiratory distress syndrome, what kills the patient, was actually shut down in the treatment group. And this is what's so sensational and promising. And now we also studied the mechanism of action. We know the different pathways that have been attenuated by this treatment, and all of this will be published shortly. So when we looked after 24 hours and we compared to animals before the procedure, this is a normal tissue histology at baseline of the heart, the liver, the lungs, and the kidney. You can see what happens. We look at the heart, total devastation of the tissue and structural damage of the control, really basically no change in the heart, in the treatment group, from one treatment. Same with the liver. Look at the difference in the lungs. You have some congestion, but you have functioning lungs. And look at the kidneys. You see there is almost totally normal, no structural change, and then here you can see the swelling of the different glomeruli. So major structural damage, this is what you call multi-organ dysfunction syndrome. This is what kills sepsis patients and makes the survivors with illnesses for the rest of their lives. So our focus is initially on sepsis. This is our projection. There's no guarantee, of course. We need to capture a very small piece of the market, 2% of the USA sepsis market, 22,000 patients, to really be a very, very, very large company. And of course, if we can capture a larger part, then you're talking about a very large company. But again, it's an investment, it's early stage. And our team, our core team, people experienced with management, I'm dedicated to this project right now. Anat Ternet has been with me at Eugenics and here, and Glenn Cherto was the head of Stanford and UCSF nephrology, who helps us with medical affairs, and a lot of additional consultant researchers and regulatory people who are helping us. We have a very strong scientific advisory board in the field of apheresis, in the field of acute kidney injury with Glenn and Ron, and in the field of oncology and immunotherapy with Daniel Kreitzman, who was the principal investigator in the famous Spectasol trial. So our training, we are going to the intensivist, we're going to the nephrologist. I just came back for presenting our research in an important sepsis conference, critical care conference in China. And we're going to focus initially on USA and Europe, and then move to other markets, Asia, Canada, Australia, Israel. And really, we have developed a very robust IP protection. People often joke that it's my expensive hobby, getting patents. So we have 60 patents, potentially even more than this, across 4 patent families covering 34 countries. So the basic patent, as long as we remove some galectin-3, no matter how, we can remove anything else. We can combine with medications. We can remove other galectin, TNF-alpha receptors, that's specifically for cancer treatment. We have patents on putting different columns in the sequence and combining them, and individualizing column. And we have patents for combined treatments for photopheresis. And we also have patents for specific indications for viral infections, including COVID, removing it, and for personalized columns, and for other indications that I can't disclose it. As from the point of view from the business perspective of the exit of the company, with potential buyers who routinely buy technologies, we are in touch with some of them for quite a few for quite a few years. We have collaboration partnerships. I mentioned Terumo. There's one more very large company which I can't mention because it's private, and collaboration and licensing, especially as we can show the role in immunotherapy with pharma companies. So the reason why this is a special opportunity to invest in our platform, it's a promising technology. We have encouraging proof of concept results. We have high safety, easy adoption. It's a technology platform. We're not dependent on one indication, and if it doesn't work, it's gone. There are so many indications, and the time to market is quicker than any drug. And it's really, we are really bringing the message that critical conditions share a single solution. So we're inviting you to join us to leverage in this breakthrough technology. That's why we are going out for crowdfunding through WeFunder. In addition to talking to big investors, and we've raised significant money already. I really, as part of my philosophy, I believe in creating community energy around this. Of course, we would appreciate large investment, but even as small as $250, that's the minimum. And for the early birds, for our community, we've done a special pricing. So let me explain to you a little bit. This is it's in a safe form, so which means that it's an investment at a certain maximum price that gets converted once we issue shares for the first time. And the pre-money, the highest valuation that the investment gets converted to share is 49.5, even if it's 100 million or 200, it doesn't matter. And if it's under 49.5, then there is still a 10% discount. But what we are doing for the early bird is we are reducing by 20% the cap. So the valuation cap now is only 39.6. So I invite you to be part of this life-saving innovation and join me in our fight against cancer. That's a link that we'll also send you. And I want to thank you from the depths of my heart for listening, for taking your time, for your trust, for your interest, regardless if you really invest or you don't invest, really for taking the time. And if this is of interest to you, please also feel free to share it with other people. It's really a community effort. I've put a lot of years, a lot of time. I changed my life plan. I was planning at 60 to sell Econogenics, which I started selling, and now focus on meditating and teaching, just sharing and educating people. But this is too important. It's important enough that I changed my life plans. And I'm totally into it. Thank you very much. Really, I really appreciate it. And I really thank you for taking the time to listen to my journey and my focus in life right now. So be well.